Good afternoon. I'm Moshe Weinstein, CEO of Everads. Thank you to the organizers for inviting me to present. I'm glad to have met some of you yesterday in person and apologize that I couldn't be here today. Before I get started, I do believe that we owe a debt of gratitude to Clearsight and Dr. Chula, as without their work, suprachoroidal delivery would not be where it is today. We even made it to prime time at OIS, which is a notable achievement. Here are my disclosures on an employee and shareholder at Everads, and as a staunch believer in this route of administration, I do also own 500 shares of Clearsight. I won't go into all the background about suprachoroidal delivery, though I do want to show this slide as it highlights the increasing interest in suprachoroidal delivery. This slide was pretty bare 2 years ago. However, despite the increased interest, the number of delivery technologies available is very limited. So Everads is enabling more effective and durable treatments for retinal disease, leveraging our novel delivery technology that enables broad and rapid distribution throughout the suprachoroidal space. Before I share how our technology works and show some videos from the ongoing clinical trial, I wanted to first give a high-level summary of our achievements to date. On the technology side, currently we're in clinical trials where we continue to prove the safety, effectiveness, tolerability, and ease of use of our delivery platform. Data from our first cohort was recently presented at Retina. This study comes after validation in over 200 non-human primates, delivering various therapeutic modalities. On the gene therapy side, we have shown that with our technology, transduction is achievable in NHPs throughout the posterior segment. On the business side, we continue to offer target and modality-specific licenses and are also developing an internal pipeline. Over the summer, we signed a license deal with Kriya Therapeutics, which earned us an upfront payment and potential for millions in future milestones and royalties. We have 2 other undisclosed option agreements with pharma companies, and we continue to develop our JAK inhibitor program with Viva Vision, and we'll highlight some of that data today. This slide presents a snapshot of our internal and licensed partner pipeline, which is all supported by the clinical study that Everads is running. Not surprising, many of our collaborations are in the gene therapy space. So what is the Everads injector? Our injector is a single-use device designed to work with a standard Lurlock syringe. The concept of the inventor was to enter the eye tangentially, and in order to not put anything sharp into the choroid, use a non-sharp tissue separator to perform a blunt dissection between the sclera and the choroid, opening a path into the suprachoroidal space. The needle of the injector has a sleeve stopper which controls the entry depth, after which the lever is pressed to extend and retract the separator, and then you inject. Simple as that. You can see here in the illustration how this technology works. We approach the eye tangentially up to the sleeve stopper, extend and retract the tissue separator, and then we inject. It's important to note that the tissue separator is not a cannula, rather it retracts into the needle and the injectate comes out of the tip of the bevel, and then you can see the distribution throughout the eye. Our ongoing clinical trial's primary aim is to demonstrate the safety and effectiveness of our injector. In this study, we're injecting 100 microliters triamcinolone in patients with DME. You can see in this real-time video of the injection procedure that is being done in an office setting. The patient is under topical anesthetic only. The physician approaches the eye tangentially at about a 40-degree angle, inserts the needle to the tip of the sleeve stopper, presses down on the tissue separator to extend and retract it, and then pushes down on the plunger, and then injects. We leave the injector in place for 5 to 10 seconds, and then cover it with a cotton tip and remove it. We also have in our clinical trial used thermal imaging to confirm delivery. And you can see here, right as the physician is injecting, a darkening area forming under the posterior to the needle tip. This is a result of the injectate cooling the sclera from underneath and confirms delivery into the suprachoroidal space. I do want to highlight some non-clinical data that we have generated. This is distribution achieved with 100 microliter of ICG in vivo in a cyno-NHP, with the eye removed and dissected 10 minutes post-injection. You can see the ICG through the sclera, which is quite translucent, and via dissection, we confirm a clean vitreous. Our NHP data was also published recently in TVST. On the gene therapy side, Everads injector was featured in 2 posters this past ARVO with 2 different serotypes, both demonstrating GFP expression throughout the posterior pole. This second poster was one that was presented by Kriya and was one of the data sets that led them to license our technology. We also recently had the opportunity to test our technology in dog models, and you can see here the fundus imaging pre and immediately post-injection of 150 microliter fitsidextrin showing complete coverage of the posterior pole. We do often get asked, how is this different from a microneedle approach? To highlight a few differences, firstly, we enter the eye tangentially, and using our tissue separator, we have demonstrated the ability to get a quick and broad distribution throughout the posterior pole. This approach enables also low susceptibility to scleral thickness. It doesn't actually matter if the patient's sclera is a little thicker or thinner at the injection site. We were able to show that we can inject large particles well exceeding 50 micron, as well as viscous materials. And we have proven the safety of this delivery approach of over 200 microliters in various animal species with limited to no reflux. I do also want to highlight our internal co-development program with VivaVision. We were jointly awarded a grant which helped us fund this initial work. For those of you who don't know VivaVision, they're an ophthalmology-focused company founded by ex-Allergan and Bausch & Lomb scientists with innovative drugs entering phase 3. As we all know, inflammation is implicated in uveitis and also in the pathogenesis of other retinal diseases. We can all agree that there is a need for potent, localized, durable, and safe non-steroidal anti-inflammatory drugs. JAK inhibitors have proven anti-inflammatory mechanisms of action and are being studied in uveitis via systemic administration. We're developing a novel, potent, and selective JAK1 TYK2 inhibitor optimized for durability via suprachoroidal delivery. This is a prodrug based on the active moiety of VVN461, which is entering phase 3 in a topical formulation for anterior uveitis and post-operative inflammation. We show here the durability via suprachoroidal injection with concentrations well above the IC50 level at 4 months so far, with no safety concerns seen via imaging. The previous clinical experience and proven mechanism of action of this molecule significantly de-risks this program, making it an attractive investment opportunity. Lastly, I invite biotech and pharma companies in the room who are interested to test our technology to reach out. We are always looking to collaborate, and we are far from having licensed out all the rights for our technology. For investors, please join our $8 million financing round and be part of our amazing journey as we further develop our internal JAK program and our licensed collaborations. Thank you very much.